Bom, pessoal, boa tarde a todos, uma vez mais, então, para nos assistir aqui no nosso novo seminário de hoje. Então, hoje teremos a presença da doutora Margarita Brandt, que vai falar um pouco sobre Galápagos. Eu quero agradecer muito as pessoas que já estão aqui ansiosos, esperando essa oportunidade para ver essa palestra, e a Margarita por ter aceito participar desse espaço para falar de ciência. Então, Margarita, muchas gracias por aceptar. Es muy chévere poder tenerte en este espacio para que hables un poco de lo que están haciendo en Galápagos. Creo que es muy chévere y puede ser una oportunidad interesante para crear nuevas asociações ou lo que se genere a partir dessa charla. Então, pessoal, a palestra vai ser em inglês para facilitar a comunicação para todos nós. E, de novo, todas as perguntas que vocês tenham podem ser colocadas em espanhol, português, inglês, e a gente faz uma discussão mais dinâmica com a Margarita no dia de hoje. Então, Margarita, aqui, thanks again for accepting this space for participating in this moment, and the floor is all yours.
Thank you, Juan Pablo. Hi everyone, my name is Margarita Brandt. I'm an Ecuadorian researcher. I actually studied applied ecology here in Ecuador. And then I went to live to the Galápagos, where I fell in love with the marine life, especially with marine invertebrates. And so you will see that basically I've been working with marine invertebrates more than 20 years. And they are, my motivation is because they are indicator species. They respond to the environmental temperatures, and that's why I think they are really interesting, especially now that we are facing climate change. I did my PhD at Brown University in the United States, and since 9 years I've been working as a professor and researcher at Universidad San Francisco de Quito here in Ecuador. I teach both here in the Galápagos classes on marine community ecology, and that's exactly what I do. I do species interactions, again, especially with invertebrates. But now, as I'm going to talk to you in this talk, the Galápagos are very special. So to ask questions about climate change and how species are responding to that. And so that's basically the focus of my talk today. So I'm gonna share some of the work that I've been involved from a long time ago, and then I will finish with the most recent one, so you can have an idea of what I've been working in the Galápagos. Also, okay, so to begin, I'd like to start with this quote from Darwin that talks about the Galápagos. Considering that these islands are placed directly under the equator, the climate is far from being excessively hot. So we are right in the equator, so we were supposed to be, like in a tropical location, typical tropical conditions, but it's not the case for the Galápagos. So the Galápagos are immersed in these complex oceanographic settings. You see here basically 4 water fronts or water currents that actually make this archipelago very dynamic in terms of thermal regimes. Basically, we have one very warm and nutrient-poor current, which is the Panama current that comes from the northeast. It basically influenced the northern part of the archipelago with those conditions. And then we have 2 main upwelling currents that brings cold water from the depth and brings nutrients to the surface and replaces those warm surface water. One is the Cromwell current that runs from the west, and the other one is the Peruvian oceanic current that, together with the Humboldt current from Peru and Chile, brings again an upwelling to the Galápagos. And so this creates conditions of different temperatures, not only spatially, as you can see here, with the northern being more warm and the west and the southeastern a little bit cooler, but also through time. You can see here the sea surface temperatures across in 1 year, and so you can see that the beginning of the year, from January to July, more or less, we have the warm season in the Galápagos, and then the rest of the year is the cool season. And I like this graph, although it's a little bit old. It's because you can see here how the temperature varies, especially in different parts of the archipelago. We have different mean temperatures, but also seasonality. And so that creates the perfect laboratory to ask questions about climate change, because we have a natural gradient of temperature. And so upwelling is very interesting and important and relevant to the Galapagos, because that's why it creates conditions that are more temperate-like instead of just completely tropical. And so that we are like on the same page of what upwelling means. We have different conditions or different types of upwelling in Galapagos. One is the continental South American upwelling that is basically produced by the trade winds coming from the southeast, and it's removing the surface waters, warm surface waters, and that creates like a vacuum. So the upwelling current actually can replace those surface water and brings nutrients from the depth. So the normal conditions in the Galapagos are actually upwelling conditions. Of course, we have this seasonality, but the trade winds are pretty active all year long. While when the trade winds weaken, we have a Nino condition, which is the opposite. So basically, there's no need for the cold currents to come up and replace the surface water. And so the conditions are warmer, heat accumulates, and we have some consequences of that I'm going to talk about. So if we take a look at this upwelling concentration through time and also in space, this is a chlorophyll A satellite images, and you can see that the west of the archipelago over here is basically considered the most productive site or influenced by upwelling. However, you can see also that in the center islands, in some other places, also chlorophyll is accumulated. And so we wanted to understand how does upwelling actually influence other parts of the Galapagos Marine Reserve that is not necessarily on the west. And so this is basically the work that I did with my former PhD supervisor, John Whitman from Brown University, and with Franz Smith, a colleague from a long time ago, that we wanted to understand how does upwelling actually create these conditions in the central archipelago. And so we chose to work in 12 sites located in the central archipelago, and so we characterized and we measure upwelling in all those sites. The picture or figure that I'm showing you here are the 12 sites based on a similarity analysis in the current flow of the equatorial undercurrent, in other words, of the Cromwell current. And so in terms of the high vertical velocities, the high percentage of time that flows was due to upwelling, and the high incidence of the Cromwell current, we characterized these 12 sites into high, medium, or low upwelling. So basically with a similarity analysis. But when we plotted those similarities of upwelling, and basically how are these sites characterized by upwelling, we can see that they are like pretty much like located everywhere. You can see in green the strong upwelling sites. That means that the persistence and the intensity of the upwelling currents are more persistent through the year. They are located like pretty much like in very different parts of the central archipelago. Then we have the blue, where these sites are like receive intermittent upwelling, and then we have the red sites that are characterized by low upwelling regions. And I want to point out here that in very small scales, for example, in Floriana, this island in the south here, we can see like very contrasting conditions in relation to upwelling. So the east, in the northern part of the island, is characterized by either low or intermediate upwelling, and the west, like this place, La Botella, is high upwelling. So in less than maybe like 40 kilometers, we have very different conditions. And so how does this upwelling translate or influence the food web dynamics in the Galapagos? So we realized that just by looking at these communities that these high upwelling sites were characterized by a lot of barnacles. Okay, and so we deployed these recruitment planes in 3 consecutive years to measure barnacle recruitment. And so pretty consistently we found that the highest recruitment of barnacles were associated with high upwelling sites. And this was very interesting and kind of like different from other parts of the world, from, for example, from the west coast of the United States, that it's actually the opposite. When a whaling actually relaxes, barnacle larvae recruit in those coasts. And here in the Galapagos, we were looking at the opposite trends. And not only barnacle recruitment was highly correlated with this high upwelling site, but also they survived into the next cohorts, and so the adult barnacle populations were also abundant in high upwelling sites. And as expected, if we are receiving a lot of nutrients and currents, that brings a lot of particles into the water column, we also were able to test that barnacle growth rate was actually strong or higher at strong upwelling sites. Consistently, all these high upwelling sites characterized by upwelling are also having high productivity. And in relation to this coupling between predator and prey, we also found that barnacle recruitment and the barnacle cover of the other barnacles was highly associated with the whelk abundance. This whelk is a predator from the barnacles, and apparently they are tracking their prey in these high upwelling sites, and so their abundances were highly correlated. And so we also did some experiments to test predation intensity across this gradient of 12 sites in relation to upwelling. And so we had some experimental plays where either we gave access to predators, or we prevented the predators to access the other barnacles, and we measured basically how many barnacles were eaten per time. And we found that the high upwelling sites presented more predation than the low upwelling sites. So in summary, upwelling in the central archipelago creates these bottom-up and top-down effects in the Galápagos reserve, because we were able to provide evidence that with high upwelling we have more productivity, more barnacle recruitment that translates into high abundance of other barnacles that actually attract their prey or whelks, and then the top-down coupling, meaning that the top control of the whelks into the barnacles were also coupled along this mesoscale of upwelling. So that was my first, let's say, experimental work in the Galapagos a while ago, in trying to understand how upwelling does create these conditions in the Galapagos. And I already told you what happens when upwelling is reduced, or when the intensity of upwelling is weakened. And we have, of course, El Niño southern oscillation, which probably you are familiar with, but I just want to highlight that it's both an oceanographically and atmospherically driven phenomenon. So both systems have to be coupled, so El Niño event can actually occur. And this graph is showing us basically the ENSO events that have happened in the region through time. So the peaks on top of the line are the El Niño events. And we also have La Niña events, which are the opposite. So El Niño, warm conditions, nutrient-poor conditions, really prolonged. And the La Niña is the opposite, like cold conditions with a lot of productivity. And you can see that two El Niño events stand out here, the 82-83 and the 97 and 98. But you can see here that the system is really dynamic. So with those El Niño events, we perceived a massive death of corals in the Galapagos. During the first El Niño, basically we lost most of the coral cover along the archipelago at 14 sites. However, something interesting happened, because during the subsequent ENSO, the strongest, one of the strongest, the 97, 98, there was relatively less bleaching than it was predicted, compared or based on the bleaching and the coral loss that we experienced in the 80s. And so these questions of, are coral populations more heat tolerant, actually came up to our minds with some colleagues from California State University, Monterey Bay. Basically, do thermal sensitivities vary among corals from different regions, considering that we have this temperature gradient? If so, which coral populations are more tolerant in both to heat and cold? Because we saw that these communities are immersed to both heat and cold extreme events. And if there's a trade-off between heat and cold tolerances, for example, if corals that are more heat tolerant will they lose that tolerance to cold events and vice versa. And which coral populations have a wide thermal trend? Oops. Um, and another question that I'm not going to talk about that we are working on this is what are the mechanisms underlying thermal tolerance? And for that, we are doing like gene protein expression of the corals and also trying to characterize the symbiont types of the corals. But that's a work in progress that we have not results yet. So this work, as I said before, has been done with colleagues from California State University in Monterey Bay. My main collaborator is Sharon Logan, with whom we designed this study that I'm going to show you, and of course other collaborators from other parts. This map is basically showing what we lost in terms of coral communities or coral reefs in the Galápagos. So the red circles is either coral communities or structural reefs that we lost, and the greens is whatever is left. So basically, we got left with a lot of coral communities. That means like rocky reefs with coral patches, and the only structural reef that is now alive is in Darwin Island, in the far northern part of the archipelago. So we lost a lot, and we were lucky enough in the 2019, I think, to go to each one of these islands where the coral communities still were present. And so we collected a coral fragment from all these islands, but I'm going to show you the results of those collections of only these 5 islands. And with this map, I want to highlight that these islands ideally are going to present thermal regimes that are very different. Darwin and Wolf, subject to warmer conditions, Isabella on the west to the coolest or coldest conditions, and the other 2 islands like a mixture of the 2. And so we had this coral reaching autonomous stress system, which is a portable a system that we can try the coral sensitivities. And we ran these experiments along these 5 islands. Even we also ran this on the boat. We had 2 tanks per temperature, and we had 2 coral fragments per colony. We had 8 colonies per island or per site. And we have a range of temperatures that we tested the thermal tolerances of the corals. So these are the preliminary results, and I'm showing you here the results to the heat stress, because we put these corals into both heat and cold stress, and we use 2 types of corals. Pavona clavus, which is a massive growth coral, and Posilopora. We had several species of Posilopora, which is more a branching one. And so we also wanted to compare how different types of growth corals were also responding to these heat extreme events. And over here on the x-axis, we have the 5 different islands or locations that we collected the corals from. And as the response variable, we have the change in photochemical efficiency of the zooxanthellae. So basically what we are looking at here is that for Pavona clavus, the corals that came from Isabella from the west are the most heat sensitive, because the efficiency of the zooxanthellae really dropped at highest temperature. And for Silopora is very similar, although the significant differences were found at not the highest temperature that we use for the assays, but at the 34 degrees. But the pattern is very similar. So the corals from Isabella for Silopora were also the most heat sensitive. Now, when we take a look at the cold stress assays responses, we were really surprised to see these results, completely unexpected and opposite that we were expecting. So these are coldest temperatures that we put these corals to this range of cooler temperatures. And we were expecting, okay, maybe the corals that were on the west or close to upwelling currents were going to be more cold tolerant. And it was the opposite. If you can see the responses of the blue are Española, which Española is this island over here on the southeast, and it has some varying conditions, but we were really not expected to see that they were like more cold sensitive, especially in relation to, for example, the corals from Darwin Wolf that came from the northern part of the archipelago that actually is subject to warm conditions throughout the year. So we completely had these different results. And this is another way to see the same results are the tolerances by colonies. We have 8 colonies per site or per island. And this is for the results for Pavona clavus. And basically what we are looking at here is there are trade-offs between the heat and cold tolerances. And for example, for the Isabella corals, we see that a maximum of 4 colonies had either medium or high tolerance to heat. However, most of the colonies, like up to 8, they had tolerance to cold. Española also is the same. It has trade-offs, but on the opposite direction. So most of the colonies, 7 colonies, were heat tolerant, and only 4 were cold tolerant. And you can see like the corals from other islands like Darwin, Wolf, and San Cristobal showing a wide range of tolerances, which was in a certain way also kind of like unexpected for us. So for the Posilopora coral was also the same patterns of the results, but we didn't see any trade-off between heat and cold tolerances for Isabella, but we did see for Española, and again for Darwin and Wolf and San Cristobal, basically we saw a wide range of tolerances because they were both tolerant to heat and to cold extreme events. So that was the first assessment of Galápagos coral thermal tolerances that has ever been done. And we were able to determine that they are thermal sensitivities and that they vary across these regions, different parts of the archipelago. How do they vary? With Isabella corals being most heat sensitive and the Española corals being most cold sensitive, sorry. And we saw again these trade-offs for Isabella only for the massive coral, Pavona clavus, and for Española for both species. And we found this wide thermal breadth for the corals coming from Darwin, Wolf, and San Cristobal. So this other figure is showing you again the thermal stress event through time with an 82 El Nino and 97 Nino being maybe the strongest warm events in the Galápagos. But and with us, of course, we have experienced, as I said before, bleaching in the archipelago. But how have species responded to this? Have they responded differently because maybe they have different tolerances? And so we see, as in other parts of the world, that species do not respond equally. So the Posilopora corals were very abundant in the northern part in Darwin. They are not as abundant as before, and Porides, another massive coral, it's becoming the most dominant. So we are looking at new losers and new winners with these climate change or ocean warming events. So now, talking to other community changes and not only about corals, we have also seen changes through time, especially after El Nino of 82. Before, there were like very few turfing algae, more abundance of brown algae, and more abundance of sponges. And after 82 El Nino, we saw that an increase in turfing algae, again general as in other parts of the world, and a decrease in brown algae and sponges. So we are seeing that species are responding differently to these extreme events. So the next questions that we are now focusing with other colleagues is how does thermal sensitivity vary among species? Does it vary among functional groups or with species traits? And how will climate change alter the species and the trait composition of the marine communities? And so with that, this is work that I'm basically doing with my main collaborator, John Bruno from UNC Chapel Hill, and of course with a lot of people who have helped us run these experiments. And so what basically we are looking is at different species, how different species are responding to these different temperatures. And so for that, we have seen we have like done experiments of thermal sensitivity with 26 species belonging to 10 phyla coming from 5 different sites, from both intertidal and the subtidal. And we are doing these experiments in San Cristobal Island, which is this island over here on the easternmost island, and where my university has a campus and a research laboratory. And so we did these ecophysiological experiments where we put the critters here in this cooler, and we do temperatures with subject these organisms to different temperatures. For algae, we had like a range of temperatures from 14 degrees Celsius to 42 degrees Celsius, and for invertebrates, we went a little bit farther because they were usually more tolerant than algae. And so we measure gross photosynthesis for algae and also respiration rates. And so we wanted to take a understand how do these thermosensitivities vary by species. And if you are not familiar, what we are trying to look is to estimate these thermal performance curves for each one of the species, which has some important variables. One of the most important variables is the temperature, the optimal temperature. That means the temperature at which any physiological rate, in this case respiration or photosynthesis, is going to be maximized. And then of course we have the activation energy, the deactivation energy, and the minimum and the maximum temperatures at which the enzymes need to work. And a typical Tpc curve looks like this, where we have the temperature that we manipulate in these experimental respirometer chambers, and of course the response variable, in this case the respiration rate. So we have an increase, a thermal optimum, and then a decrease. And so we measure that for those 26 species. However, we only end up with 17 results for 17 species. And after that, or together with those Tpc and respirometry experiments, we also classified each species based on the phylum, the reproduction type, the habitat that they came, the size, and in total we determined 16 traits of those 17 species. And so what you're looking at here are these estimates of the Tpc curves for the 17 species that we ended up results with. And I would like you to focus only on maybe the thermal optimum, the TO that I just showed in the Tpc curve. And you can see that there is a lot of variability. There are species that have high thermal optimums and other species that have low thermal optimums. And of course, that's expected that the species are going to respond differently. So one way to take a look at this with one trait, for example, if we take a look at the functional groups that the species belong to, and we compare the thermal optimum and also the CT max, which is the maximum temperature, we can see that there are some significant differences. So primary producers are the most sensitive because they have the lowest thermal optimums. And so if we continue to see this increase in ocean warming, and species respond differently, of course we are going to see some changes in community structures because maybe the primary producers are not going to keep up, right? While others, maybe they are going to be a little bit more tolerant, or maybe they are going to feed more on primary producers if we are talking about herbivores. And so with this is something that we are looking at with other experiments that actually some species respond differently to these warming events. And over here you see community development of macroalgae in the peak of the cold season that I show you in the Galápagos in July, August. There are some species that come that are very abundant, but as soon as it gets warm naturally, because of the seasonality, they just leave. They don't tolerate the warm conditions. And we are looking at the same results with different experiments. And this graph is the last that I wanted to show you about this experiment. Is basically the 16 traits in a multidimensional analysis of the 17 species. And what we are looking at here, and based on their thermal optimum. And we are looking at here that they group each other based on the similarities of their sensitivities, but also on their traits. And the ones that are the most sensitive, right, are the macroalgae, this group here, that have the lowest thermal optimum. And we can see that filter feeders like sea anemones and sponges, they also group here. They have like intermediate thermal optimums, and then the rest of the species, which are either interstitial, like this crab and so on, they also have a different set of traits that are also responding to these different warming events. So it's really interesting to see this community approach because species will respond differently to the warming. So does this imply that we are going to see changes in composition and ecological communities? Probably yes. We have seen other studies that have already seen these changes in functional group abundances or identities that were mostly attributable to warming events. But of course, we are just in the Galápagos, we're just beginning to explore this and to understand. We have a bunch of species to work with. And we are just beginning to understand this. And just to finish, I also would like to talk about another published work that we did with John and Nisa and Isabel about these thermal performance and thermal sensitivities, which adds a little bit more of complexity to the system. So Isabel, she did these thermal performance curves with the pencil urchins, this urchin that is really, really abundant and common across the whole archipelago. And she collected the urchins from 4 different sites that are subject to different temperature regimes. And here you're taking a look at the Tpc curves of 4 different populations. And one thing that it really stands out is that the Tpc curves are very different depending where the urchin populations came from. They have different thermal optimums. So the coolest site, which is La Botella, this one has a thermal optimum of 30 degrees Celsius, while Bartolome, the warmest, is about 33. And of course, we can see that some populations are really at their limit of their thermal performance curves because they, as soon as they experience warmer conditions, their populations crash. And so I wanted to finish the talk with this slide because exactly this is what it makes the Galapagos so interesting because the conditions vary through space and time. And so we can not only try to understand how species are responding to warming events by just looking at one population in a single place in the Galapagos, but also from different regions that are subject to different temperature regimes. And that is also good news because some populations can be, acclimatized or can have, the option of acclimatate, in, while others, as we can see here, are probably going to have their limits, and that's really interesting in order to predict how communities could change with time. And so, with that, I would like to thank you for your attention, and I will be happy to take any questions.
Uh, okay, thank you, Margarita, for your excellent talk. It's very nice see again your presentation and learning more about the process in Galapagos and of this specific experiment you can do in these islands. Well, pessoal, então agora vamos abrir o espaço para as perguntas para a Margarita. Acho que ela trouxe bastante coisas bem interessantes para a gente discutir aqui. E, de novo, podem escrever perguntas em português ou em inglês para ela diretamente. A gente tenta fazer uma tradução. Margarita, al lado derecho está la parte del chat y ahí está el chat general que conecta. Entonces, si hacen alguna pregunta en inglés, puedes como mirarla. Y yo intento leértela, pero pues igual si no me entiendes, está ahí para facilitar.
Super, muchas gracias.
Vale, the first question is more one curiosity I have about the reef fish, and you're working so much in Galapagos, and I remember the last Nino phenomenon very strong, I think 20 years ago, and one only endemic species is disappeared totally, is Asura.
Epalina.
Yeah, exactly. And this species is very particular because it's endemic species and never more see it. And you see these similar effects in another type for the species in invertebrates or another type for the invertebrates.
Um, we have really not, registered like other species laws in
Hmm.
addition to the ones that you said. There are some macroalgae that actually also disappear from the intertidal habitats, and like the Bucaria galapagensis that was kind of common in the central islands. But like the one that you just mentioned, we have really not documented before. As I said before, with the corals, we have not experienced any local losses or local extinctions, but just the cover of the corals that we have lost. And as I said before, it seems that the relative dominance of the species is also changing. But yeah, and I cannot think of another example of a loss because of a strong El Niño event.
Hmm. Well, another question I have: when you lost several species, coral species, you lost the rugosity or another type for the complexity for the habitats, but you can see it or you can talk about the change in the community structure for another level. For example, with fish, invertebrates, his change or not. What is the observation you have in this in Galapagos about this point?
So that's a really interesting question, and this is something that we were talking about with a colleague that with whom we started a coral restoration project in Isabela. Is like, what else have we lost? Not only the coral cover, but have we lost also the communities associated to them? So the strongest El Nino was in '82, right? It was a long time ago, and so the systematic information, like surveys or monitoring, especially done by the Darwin Foundation, um, started in 2000. So, like a real picture of how communities were before that El Nino event of '82, we don't have that. Like a community vision of the abundances and the species. But I'm sure that definitely with the loss of rugosity and complexity in the habitat that the corals provided, we lost many species that we were not aware of. Um, eh, but I don't have an answer to your question. But probably, and I know also some colleagues from the national park are doing also some restoration projects in the Central Archipelago. The idea is that we are going to measure that, to measure those coral communities that don't have much coral cover in the future. Hopefully, with the restoration project, we are going to bring back, I would say, like more diversity or other species that are going to use the coral um, reefs for habitat and so on.
Nice. Okay, I've another question is, well, the people in the world explains several points in the transition zone. You can see different tropicalization case when you see different tropical species extending your range of the distribution. But I don't know, Galapagos, you have seen Floriana is very stable because you have the old wedding and up time in this part. But the temperature increasing all the time. And the point is, you start to see more tropical species in this space when you have more upwelling or not.
That's also an interesting question. And I don't have the answer to that. No, I mean, apart from what I gave in the talk, that we after a new event there was like this community change from few turfing algae to a lot of turfing algae. I mean, that's one sign of the tropicalization, right? Like these
Hmm.
turfing algae that are really fast growing in that they are benefited from warming conditions. We see this pattern everywhere. Although I don't know. I mean, I'm not tempted to say that some regions are. getting tropicalized. I wouldn't dare to try to answer that because again, you need like a very long-term monitoring and have like a really good idea of what was before to see if
Mhm.
actually any changes are actually related to warming. Another thing is that we don't know if these high upwelling sites are actually becoming warmer or not. I mean, the study that we did with John Whitman was like in the first 10 years of 2000, right? And so it was basically one of the few studies that tried to characterize upwelling and the ecological dynamics within a gradient of upwelling. But I'm not really aware of any other researchers that are trying to see how this intensity of upwelling in the central archipelago is changing with climate change. That would
Mhm.
be really interesting because that would be like one way to correlate, how does if upwelling intensity is changing or not, and how does these communities or how do these communities respond to this warming if they are actually promoting tropicalization or not. But it's really interesting.
Yeah, nice. Well, this is my last question. Is more one another curiosity about your result and your experiment for the acclimatization in the different organisms. But many species in all the generations produce more resistance for the different stress. For example, in this case, temperature. And the small scale, you see this type for the acclimatization for the new generations in invertebrates, for example.
In barnacles, did you say?
Yeah, in any invertebrate, you start to measure the thermal tolerance.
Um, I don't have the answer again for that because I mean, so we are just basically what we are trying to understand with these different thermal performance curves with different
Hmm.
species is something that we think is acclimatization that doesn't really involve any evolutionary response, yeah, because we are just taking a look at one population and at a single period of time. And now next year we are going to see the same species, or we're going to evaluate the same species across different seasons to see also if the seasonality has anything to do with that. But to answer that question that you make, we probably need to do, as you said, with different generations of the same organisms
Hmm.
to see if actually these evolutionary processes confer adaptation and not necessarily acclimatization. And this
Okay.
is something that we have not done before. Um, with the coral project with my colleagues, we are hoping to try to understand that because we are doing genetics with that. Although we would need to repeat again the same experiments in a few years or something like that to see if there's actually any evolution going on with these populations in relation to their thermal tolerances, and not only of the corals itself, but also with the symbionts that they are having.
Hmm. Hmm.
Mm.
Nice, very nice. I think your work in Galapagos is very nice. This is perfect for different research for the climatic change, I think. Pessoal, alguma pergunta para adicionar ou que vocês queiram, algum comentário para a Margarita? Acho que é um momento ideal para a gente pensar um pouco mais sobre a nossa realidade no Brasil, comparativamente com o que está sendo visto em outros lugares, por exemplo. Bueno, Margarita, entonces, pues nada, muchísimas gracias nuevamente por participar. Creo que sigo insistiendo, es muy interesante trabajar en Galápagos, es un perfecto lugar, laboratorio natural por naturaleza. Y nada, espero que podamos seguir conversando y poder sabiendo mais sobre tu trabalho em Galápagos. De qualquer forma, pessoal, eu coloquei o link do laboratório de Margarita e quem tivesse interesse ou alguma pergunta particular pode entrar em contato diretamente com ela. Toda essa informação descritiva está na informação do YouTube. Quem quer assistir ou ver de novo para poder entrar em contato com ela em termos de parcerias ou qualquer dúvida que seja gerada a partir de hoje. Então, também quero aproveitar então para convidar a todos na próxima quinta-feira para a gente continuar nesse programa de CEBIMários que a gente tenta trazer cada semana aqui. Então, Margarita, muchísimas gracias. Thanks again.
Gracias, Paula, por la invitación. Justamente puse en el chat mi correo electrónico por si se les ocurre alguna pregunta después de la charla, que me puedan escribir. Si tienen...
espérate, lo has puesto en el chat el privado e ya lo voy a colocar en el público.
Buenas.
Pessoal, sim, coloquei então o e-mail da Margarita no chat para todos poder ter o contato dela e aí ela fica à disposição para conversar com vocês, quem tivesse alguma dúvida. Então, nada, Margarita, muchísimas gracias y seguimos en contacto.
Listo,
Cuídate
gracias
mucho.
por la invitación. Chao, chao.
Vale, chao.